Então, Raquel, bom, primeiramente, bom dia a todos, né? Bom dia a todos os participantes e especialmente aos speakers, né, que disponibilizaram em parte do seu tempo para compartilhar com a gente as suas experiências. Tem sido experiências muito importantes, muito relevantes para todos nós, né, que estamos ainda construindo um entendimento, né, tentando dar um direcionamento para o tema aqui no Brasil. Então, hoje é o terceiro e último módulo, né, desse tema. Como a Solange falou, foi muito relevante, muito importante, muito produtivo. Então, e hoje certamente não será diferente. Eu agradeço muito a todos vocês, a todos os speakers e desejo um ótimo evento. Muito obrigado e um bom dia.
Marcela, é com você.
Obrigada, Cláudia. Então, hoje a gente está no nosso último módulo, módulo 3, a gente vai contar com os speakers, o speaker Alexandre Lidavri, que ele é diretor sênior em epidemiologia global. Ele lidera uma equipe global de epidemiologistas que está desenvolvendo soluções para dados e evidências do mundo real. O nosso outro speaker será o Charles Lee, ele é diretor executivo de ciência regulatória na AstraZeneca, supervisiona a ciência regulatória e estratégia para doenças cardiovasculares, renal e de metabolismo. A Júlia Spinardi e o Rodrigo, que eles são da Pfizer, a Júlia é líder médica para vacinas na Pfizer Brasil, e o Rodrigo é diretor médico sênior de vacinas da Pfizer para a América Latina. E o nosso outro speaker é o Patrick Verpilat, ele é head global de epidemiologia da Merck, médico especialista em saúde pública e epidemiologia com PhD em epidemiologia genética. Acho que a gente pode começar, a gente vai começar com o Alexander.
Muito obrigado. Deixa eu ver. Bom dia a todos. Good morning. It is my pleasure to take part in the third module of this very productive workshop, as we've heard. I'm going to be talking about going from theory to practice and summarizing some of the discussions and some of the main topics that we've had related to how can we make the most out of real-world data to inform regulatory drug approvals. Okay. My disclosures are that I am a full-time employee and stockholder of AbbVie, and my opinions in this presentation are those of my scientific perspective and not necessarily those of AbbVie. And we've had a very productive 3 sessions so far, and we're entering into the third module where we will talk about specific examples. In the first module, we did an overview to talk about real-world evidence and regulatory decision-making with perspectives from agencies from the US, from Europe, from Asia. We also started talking about some of the methodological and data-related challenges and considerations. And we completed that day talking about some of the regulatory perspectives. In the second day, we talked about quality and robustness of real-world data, sharing our perspectives on the opportunities and challenges, and thinking about how to extrapolate insights from one country to other countries. We talked about transparency and data processing, and we also highlighted how registry data can be used to support some of these regulatory decisions. And then in the third day, we had a deep dive perspective into the situation in Brazil, where we reviewed some of the sources of data in Brazil, including some of the existing sources of data, such as DataSUS from the National Health Insurance System. We highlighted some of the challenges and some of the opportunities, and we shared some of the examples where we can use these data to generate meaningful insights with our presenter from IQVIA. So from my perspective, I think it's been a really interesting and worthwhile conversation, very topical, very current with the topics that we're seeing emerge across the world with the challenges of real world data. And I really want to congratulate on the organizers for pulling together great speakers and great topics to really shape the conversation in Brazil. So why are we talking about real world evidence? I think we have seen that it's growing in importance. It can be used to evaluate drug safety, effectiveness, and the value of medicines. It can also reduce the cost and time associated with drug development to get the right medicines to the right patients more efficiently. It is also an opportunity to capture the patient's voice. And I think we've seen also from regulatory agencies that clinical trials are limited and are not enough because medicines are approved and then used in broader populations that are reflected in the clinical trials. We've also seen that regulators are implementing new programs to drive the application of real world evidence with 21st Century Cures Act in the United States and Darwin EU with the EMA. And there's also been an increase in the availability of data. There's a lot more data being created, a lot more data that can be harnessed to inform some of the decisions that we would like to make that are data driven. And there's also been significant advances in the data analytic capabilities, such as with artificial intelligence. Over the last few weeks with this workshop, what we're really talking about starts with data, but then proceeds to apply the right methodology, the right scientific thinking to generate the fit for purpose evidence. And so we always have to ask ourselves when we're using real world data, are the data suitable to answer the study question? For example, for a regulatory decision. So we can't forget this. This is paramount to what we've learned over the last few days. And I think it's also important to recognize, what do we mean by real world data? What do we mean by real world evidence? And you can see some perspectives globally that are emerging from the FDA, where they state that real world data is information about patient health status and the delivery of healthcare that's routinely collected from a variety of sources. So it's a lot more open in terms of the sources. Visa has also provided some opinion about what real world data is, which is very similar. It's about patient's health or a clinical condition that's routinely collected from a variety of sources, and then they go to describe the types of sources, such as electronic health records, or medical procedure, claims data, or patient-generated data. When we look at the column for real-world evidence, it's consistent that it's essentially the evidence that results from the analysis of real-world data. And then we also saw who is talking about real-world evidence, which parts of the world are thinking about new guidance and ways to think about real-world evidence to inform regulatory decisions. Of course, there's been a lot of discussion about the FDA in the United States because of 21st Century Cures Act, where the FDA is on the hook to make sure that there are some guidances related to how real-world evidence can support new indications or post-approval requirements. But all of this conversation and evolution in the United States hasn't changed the evidentiary standard that is required for drug approvals, which was set forth in the 1960s. In Brazil, we've seen that there are some perspectives that are now available online. And in Europe and other parts of the world, we had speakers share with us how the PMDA in Japan or China or Taiwan and even Singapore are developing ways to think about real-world evidence for regulatory decision-making. We also had speakers in the deep dive in Brazil, for example, that shared that, this is just one tool in our toolbox of how we generate evidence. Every data source and every study design will have strengths and limitations. And real-world data and observational studies are one strategy to generate the right evidence. But we also have, and we were reminded that for a very controlled experiment to understand whether a new intervention has the effect to change disease course, that randomized clinical trials are still in many ways the gold standard and have to be conducted. But real-world evidence has many potential uses. And there's even efforts right now, such as the RCT duplicate, to demonstrate, can we use real-world evidence to try to replicate or have findings that are consistent with a randomized control trial. And so all these efforts and these types of case studies are important for us to take the learnings and understand where we can go with real-world evidence. Real-world evidence can be used to inform, for example, long-term safety. And epidemiologists working in companies like where I work have been thinking about how to design post-approval activities to measure the safety of a new agent after it's approved in the market. But the bottom part of this figure is really where there has been a lot more development. Can we use real-world evidence to inform a new drug approval? Because there are instances in a rare disease, there are instances where it's unethical to have a randomized trial or just not feasible, where we can leverage real-world evidence. We can also demonstrate effectiveness for new indications potentially, such as pediatrics or in oncology. And we can also use the approach for accelerated approval and then demonstrate the value of the medicines after approval towards a full approval. And we've seen the FDA provide the framework in 2018 that spoke to what they're looking for when they're thinking about real-world evidence for regulatory decision-making. And it's about the suitability of the data. Is it fit for purpose? It's about the study design and the methods. And then it's also about the regulatory considerations. Does the study meet the requirements by the FDA for data standards and the conduct of the study? We were expecting an update in December of this year to the framework, and breaking news is that last week we were provided with, for industry comment on the updated FDA guidance now. So this is now a guidance evolved from the framework just last week. And you can see from the sections of this document that it really highlights exactly what we've been talking about over the last few days in this workshop. So it's really great timing for us to talk a little bit more about this. And you can see here, it talks about the data sources to ensure that the data are relevant, that the capture of the patient journey is there, that there's an understanding of what information is being pulled forward and what is missing data, and validation work to show that the data are in fact reliable. And then comes design and methods. Are we thinking about the right time periods, the right study population? How do we define exposure? How do we define outcomes? And all the process towards validation, including for covariates that can be important confounders to understand effect modification and other contributors to the risk of disease and the risk of the patient journey being altered. And the final piece is around transparency. When you're working with real world data, the FDA would like there to be documentation about where the data are coming from, how the data is transformed and processed, so that you have a final study-specific data set that is reproducible and there are quality assurances and quality checks in place. Just to show you an example of this new guidance and what we see right now, we see again that the FDA is talking about the strengths and limitations of data sources. So we have to, when we're thinking about regulatory grade evidence, we have to provide why we chose a data source, some background information on the data source, some background information on the healthcare system that it represents, and there is specific acknowledgement around claims and electronic health records as being valuable for this real world evidence. They don't talk about registries specifically in this guidance. I think because they wanted to focus more on electronic ways that data are being generated routinely from clinical care, but registries do bring a lot of value in the ways that we can provide real world evidence, as you'll hear even today. The FDA also, again, this is another preview into this guidance. The FDA talks about describing the relevance of the data source, that it remains critical to understand if it's relevant and any bias related to the data source, such as in claims, may not capture all the information that you need to show related to a specific disease area. And when you're thinking about designing your study, you have to think about whether the data source have a representative population, have enough sample size, and if you can capture the patient journey reliably over time. Again, this is a preview of what they're talking about in the guidance, that they mention even the technologies that are available to help mine unstructured data from the electronic health records, or efforts to link across different data sets to create your study-specific data set. And all these details have to be documented and provided so that the regulators understand exactly how the data were processed, leading to the final study-specific data set. And repeatedly in the guidance, we see mention of validation studies, and it really highlights some of the challenges that we've heard over the last few days about real world data. Misclassification is a common problem. The FDA talks about providing sensitivity, specificity, positive predictive value, negative predictive value, and you can see in the table below, that's a screenshot that they mentioned that this would be something that they would look for to make sure that you can capture the right information that is needed for regulatory decisions from the evidence that you're providing. And when you're building these validation studies, it's important to also document the process and how you chose the standard for comparison. I think the importance of study design and methods cannot be understated or overstated, because the regulatory guidance it talks about these elements that they're looking for. They're looking for the design. Is it adequate scientific evidence to answer the regulatory question in the design? They're looking at the conduct. Does the study meet the regulatory requirements? Is there transparency into the process? And they're looking into the data. Are they fit for use? Are they relevant, reliable, valid? And if you're doing a comparison to a clinical trial, are the cohorts comparable? And then finally, the analysis is what they're looking for as well, that it's pre-specified, that you are controlling for confounding, thinking about bias in the source data with the methods and some of the feasibility work. So today we're going to be talking about case studies, and I think some case studies are really important to highlight what we can learn from those experiences. And I mentioned before that there is historical precedent going back to the 80s or even further back where we can use registry information or historical data, especially for rare diseases, orphan indications. And even the blinatumomab example that is more recent and is always highlighted was an approach that looked at historical patient charts to inform on what would happen to patients because the standard of care had not evolved for a very long time. But then in the bottom row with avelumab, we do see that electronic health records were used to help with a regulatory decision. And the example from Pfizer with male breast cancer, which was a rare population, that the evidence provided was very fit for purpose. For 3 different questions, there were 3 different analyses of 3 different data sets that were used to help inform. And again, just a reminder that we talked about this, that last year, that there were a number of submissions that included real-world evidence for drug approvals. But the FDA maintained that even though some of the evidence was substantial or supportive, that the basis of approvals really remains on clinical trials. But there are now 61% of the package inserts of the labels do refer to real-world evidence studies in their findings. And the FDA does talk about, external control arms and what that really means. And it's essentially a population of patients that's external to the study. But a lot of the conversation is around real-world data supporting an external control arm, for example. And what I think is also important is that we can learn a lot from the less successful, inconclusive evidence examples as well. In this report by the group Aetion, they do highlight 3 different drugs that were in these indications of multiple myeloma, bladder cancer, and lung cancer. And they talk about ways that the studies could have been successful. So for example, integrated planning of the real-world evidence external control arm at the same time as the clinical trial, having the eligibility criteria be the same between the real-world evidence arm and the clinical trial. And, things like index state definitions, to align that better between a clinical trial and the real world data cohort to avoid immortal time bias, for example. And they recommended, if possible, that trial sites can provide the real world evidence external control from the same sites. But I think one of the key learnings is that the early and frequent engagement with the regulators is really important. In Brazil, I think there's a lot of opportunity, and there are strengths, and of course there are limitations, but as long as they are well understood and they are relevant and reliable to answer a question, real world data can generate fit for purpose evidence. Through DataSeuss, there are a number of different data sets and information sources that could be used for very specific questions. as long as the data are relevant, reliable, and valid, and the evidence is consistent with the understanding of the disease with previous studies, such as from other countries or other data sources. So this is sort of close to conclusion slide, which is to say that, using observational studies and real world evidence to support effectiveness questions for regulatory decision making is making progress, but it's still a long and challenging journey. So we are on the right track. And these are the themes and the challenges and opportunities around data, design and methods, but also importantly, acceptance by researchers, by the regulators, and even within the industry of biopharma. And consistency in standards across the regions can be very helpful, and innovative and successful case studies where we can share those learnings. So how do we gain that acceptability? I think you can see from the guidance from the FDA, it's all about scientific rigor and a common understanding of what constitutes fit for purpose. And we can also move towards greater acceptability of real world evidence with the development of guidelines, guidance, and best practices through extensive stakeholder interaction like this workshop over a number of weeks, which makes it possible for us to carve out the time because it's not a full day or a full 2 days. I think the organizers did a nice job with that. Striving for internationally harmonized approaches and agreement on the standardized outcomes. And then, as we'll talk about today, the demonstration of case studies and the shared learnings from those to inform future efforts. So with that, I can hand it over to the next speaker.
Obrigada, Alexandre. Agora nós vamos com o Patrice.
So, hello everyone, I will share my screen. And I guess you can see the slides now and hear me well. So thanks, Alexander. I think that you already introduced a little bit avelumab, which is the first use case that I will present, really talking about indeed a rare cancer indication and use of real-world data, real-world evidence to support our dossier submission in this indication, which was Merkel cell carcinoma. And shortly at the end, because it's more quicker, I will say, I will also give another use case, which is more related there to off-label use in pregnancy and use again of real-world data, real-world evidence to support label update. And here in that case, it's the use case of interferon beta called REBIF, and it's used in multiple sclerosis. This is my disclosure, but same as Alexander's one. So here, just to show that indeed we are still considered, and that's definitely fine, that the gold standard for pivotal efficacy evidence remain randomized controlled studies. In practice, and we've seen this for many years now, we see also that the use of real-world data evidence for regulatory decision when it comes to safety assessment and related label updates upon request by regulators is indeed well accepted. Then the acceptability of proactive use of these real-world evidence and coming from real-world data for sponsored design label changes regarding safety and also from time to time effectiveness is less well accepted, but I will say still under consideration. And on a case-by-case basis, this evidence to complement effectiveness in initial findings may be also acceptable in area of high unmet needs, rare diseases, and depending also for sure, as Alexander said, about the quality of the data, the fit for purpose, and the robustness of evidence. So here you will see that the first use case is more referring to the bottom of this, and the second is more to the second line. So moving quickly to Bavencio and Merkel cell carcinoma. First of all, talking about the context, Merkel cell carcinoma is an ultra-rare and aggressive skin cancer. As you can see, linked to the incidence of MCC reported in this slide. And it's also linked to a poor survival outcomes and spare treatment options, because indeed prior Bavencio use and acceptability, then in stage 1 and 3, the only treatment options were surgery, radiation, and adjuvant chemotherapy, and it was even more restrictive when it was at stage 4, because in that case, in first line, you were able to use chemotherapy, and there were no clear standard treatment in second line plus. So the development we did with avelumab, Bavencio, was indeed to have a pivotal study, which was a single-arm trial designed to assess best overall response as primary endpoint. This study is referred as Javelin Merkel 200 and was targeting second line plus indication. And we complemented this single-arm trial with what we call external controls. So coming from a non-interventional or observational study based on secondary use of data from 2 different cohorts, 1 cohort in the US and 1 in the EU. Thanks to this, I will say, you will see later that we were successful to get a conditional marketing authorization in the EU in September 2017, and also an accelerated approval in the US in March 2017. The condition also for sure was to get confirmatory via a study which is still ongoing, which is named Javelin Merkel 100, this time in first line setting, which is also a single arm trial design with a duration of response as primary endpoint. So shortly to describe a little bit about what we did in terms of non-interventional study to bring additional evidence. So this study was a supportive one with the objective to provide a comparative historical reference to the single arm trial, Javelin Merkel 200, also recognizing that indeed there were very limited information coming out of the literature review that we did. The design was definitely discussed prior initiation of the study with key health authorities, so mainly the US FDA and the European Medicine Agency. We were using 2 different sources of real-world data corresponding to part A, or the first cohort in the US, using electronic health records, from an oncology-specific data source, and the part B coming from German-speaking countries, Australia, Germany, and Switzerland, which were using data from an MCC-specific registry established several years ago. We had quite some interaction, and you will see it's a little bit more detailed in the following slides, with the FDA and the EMA all along the process, and importantly also when we started to think about the study, to really discuss with them the design and how it could be used. So first of all, looking at the FDA interaction. So the first meeting took place in February 2016. And here, the feedback we got that the FDA was mentioning that it would be very challenging to set up such study. They were really advised us to have an alternative approach. And they were also clearly saying or pointed out that the data that we would generate with this non-interventional study will not be considered for comparative labeling claims. In the letter we received a month after, this was indeed again saying that they will not use this data in their review. However, in September 2016, during another meeting, they also pointed out that the results of the pivotal single-arm study will be only convincing if finally the enrolled patient will reflect the target population that we have identified in the observational setting. So what we can say is that the acceptability level was quite limited at the beginning and increased a little bit all over the discussion we had with them. Now turning to the EMA interaction. So we had much more interaction there. The first time was in May 2014, and the agency really said that they would appreciate to see good quality comparative controls. So we worked and then came back to them in July 2015 with a proposal, and they say that indeed the proposed observational study that we proposed, in fact, would allow prospective analysis that could provide good quality information and that could be acceptable for conditional approval. All along the remaining interaction, they say that indeed they request additional source to substantiate the data, because we were highlighting also that the number of patients finally that we could get out of the different data sources could be limited. The objective was indeed for us to identify the best patient as much as possible comparable to the patient included in the single arm trial to be able to make this comparison at the end. And finally, you will see in the next slide that this was a limited number at the end. But besides this, they also say that the clear criteria to match the pivotal single arm study eligibility criteria explain these limited final numbers. And this was also one way for them to confirm the ultra-rare nature of the patient population. And finally, they were asking us to clearly describe this selection process in the dossier, and also mentioning that because we were not only looking at second line plus patient, but also first line, that when we will get the results of the first line data, it could be also very valuable to put into context the results of the single arm first line confirmatory study that we would initiate after approval. So you see there that at the beginning, the acceptability was a little bit higher, I will say, than the FDA, and finally quite interesting at the end of this process. So going a little bit more into the data, that before moving to how they have received it in the dossier. So here you can see that indeed, either in the part A based on the US cohort or part B in the European cohort, then we started with quite some patients. Even if it was a rare population, we were able to get around 700 or I will say 1,000 patients in US and EU respectively. But unfortunately, I will say when you started to apply the inclusion criteria, meaning from the Merkel cell carcinoma population to the metastatic MCC population, then first line, second line, and additional criteria, then we ended with very low numbers. It was even lower, I will say, when we limited the population to the one that were really included in the trial, which was the immune competent population. So 14 patients in the US at the end and 29 in Europe. What I would like to highlight there are some results. So you can see that the objective response rates were quite different between the US and the EU cohorts, which made both agencies questioning data validity. But it's true that the limited sample size is also translated into a large confidence interval, which makes the interpretation quite challenging there. Interestingly, when we looked at the duration of response, this was quite consistent or homogeneous, providing confirmation also on the poor efficacy of chemotherapy used in this setting. And finally, when we looked at the durable response rate, then we've seen that indeed, based on these historical data, one of the most relevant endpoints, which is the endpoint that has been selected for the confirmatory trial, is definitely linked to this duration of response and durable response rate. This is a graphical representation of what I've seen before via the PFS, but you see indeed with the data of the Javelin trial, so named 00.003, sorry, and the data from the two cohorts, US second line plus, EU second line plus, put into perspective also with the publication that were released more or less at that time. So now how it has been perceived all along the interaction we had with them during the review of the dossier. And here is a copy paste of what you can find. It's completely accessible either at on the FDA website or the EMA website. So this was interesting, and I have highlighted. I will not go through all the slides, but just some of the key points that indeed finally during their when they wrote their summary of review, the FDA considered that the results of the NIS were exploratory and definitely used for clinical context. They also said that indeed there were no formal statistical comparison which were undertaken because potentially the study was also subject to selection bias, but the data were considered for informational purpose, and they confirmed in their conclusion that indeed it has provided context for the natural history of MCC with chemotherapy. And there were a podcast a few weeks after the approval of Bavencio in the US, and the moderator was asking indeed to the FDA representative if real-world data were going prime time there, because this was indeed the first use case having real-world data coming from external sources in the dossier and being part of this review process. And the FDA said that yes, indeed, a retrospective observational study was submitted to describe the natural history of metastatic Merkel cell carcinoma. And in fact, it was helped to understand the relatively poor treatment outcomes following administration of what was available at that time, meaning cytotoxic chemotherapies for treatment. In the EMA, here again, copy-paste from the EPAR that is available on the website, and you will see again that here in some of the section, it was highlighted that the data that we provided coming from the real-world data analysis were supportive data, even if not as high quality as a comparative control, which will come from a randomized clinical trial. They also highlighted that the data were showing marked geographical differences reflective of the lack of consensus regarding the therapy when it comes to these patients. And they also definitely included this in the effect table as a comparator arm. But this was not the end of the story, because in Europe, we had also an additional non-interventional study using real-world data coming also from the registry that we used as external control, but that we enhance now. So we have a post-approval commitment, which is a pass where we will assess more specifically in the subgroup of immune-compromised patients, because you may have noticed that in the clinical development up to now, we have only included immune competent. So hopefully in few years, we'll be able to confirm that Bavencio, when used in also immune compromised patient as well as immune competent patient, is effective and safe, thanks to real world data. So as a conclusion of the first use case, we can see that indeed in the first interaction with the different regulatory agencies, data were not seen as so much acceptable to replace an active control coming from a well-randomized controlled clinical trial. However, with more details, first results, discussions, the agency were more open to such an approach. So thanks to the better understanding on the disease and treatment options over time, the acceptance also increased and finally was seen as a valid option to substantiate the fact that there were very limited existing data in this context, even if, as I said, we have highlighted all the time that this was based on limited numbers, and we were also always highlighting clear limitations linked to these studies. In addition, I think that both agencies have accepted the data as supportive to help to contextualize the results of the single-arm pivotal trial, and as well when the results will be available of the confirmatory trial, which is still ongoing, and that will definitely characterize the benefit-risk profile of avelumab. Even if, as you have seen, this was not used for label claims as raised either during the meeting or during the assessment. And I think it was also used in Brazil, if I'm correct. So moving to the second use case about interferon beta and label update. Here, the context is really the use of interferon, which was, I will say now, contraindicated during pregnancy for women having multiple sclerosis. In fact, there were no evidence at all because it's pregnancy use, definitely, but there were some cases reported that were showing with inconsistent results that from time to time it could be linked to congenital malformation, spontaneous abortion, or low birth weights. So that's why we had a discussion, and I say we here, it's not only Merck, it's also other marketing authorization holders, of interferon beta, Bayer, Biogen, and Novartis. And the CHMP from the European Medicine Agency requested all of us to collaborate together in a joint path. So that's what we did. We developed and we performed a population-based cohort non-interventional study using data from Finnish and Swedish registers to evaluate pregnancy and infant outcomes in multiple sclerosis population exposed to interferon beta, but also compared to an unexposed group. The results confirm that there was no increased risk of major congenital abnormality in live birth linked to exposure to interferon beta in women with MS during pregnancy. You see there the number of pregnancy outcome and the number of women that were included into the study. And there were also no difference in the risk of spontaneous abortion, which could be another way to assess, besides MCA in live birth, the probability also to have babies with major congenital abnormality, but not viable up to the end. So no difference between women with MS exposed compared to women unexposed. The consequence of this has been an update of the different sections of the summary of product characteristics, so that in the label now, the contraindication on the initiation of treatment in pregnancy has been removed. And we have also updated the recommendation on use in pregnancy, as well as including results from this real-world data, real-world evidence study into the label. And what's interesting there, it's to explain that from time to time, it's not because data are coming from one specific geography, and here it's the Nordic countries, Sweden and Finland, that it cannot be used also in other ones. So definitely it has been used not only by Merck, but also by the other marketing authorization holders to revise label in other geographies, as you see there, including Brazil. And most probably, even if some of these regulators ask us to do additional analysis, it has been well accepted and perceived. So the different reasons may be that indeed Nordic registry data are definitely well known to have high quality data. It's also a safety outcome, so there is reasons to think that there is no impact on the way the patients are managed there. The evidence was quite robust, thanks to also a robust research plan that was discussed several times with the European Medicine Agency, and all study documentation was available on the EU PASS register, meaning before we initiated the study, predefined, which is also a guarantee, as raised by Alexander, of doing things on the right way with being transparent. It's not also not the end of the story, because now there is an additional joint pass that has been requested by EMA to address the limited experience we have still with the use of interferon beta during the second and the first pregnancy trimesters, because up to now, as it was contraindicated, it's true that most of the time the treatment was stopped as soon as the woman was aware that she was pregnant. And this is the end of my presentation.
Obrigada, Patrice. Agora a gente vai passar para os speakers da Pfizer.
Olá, bom dia a todos. Vou compartilhar minha tela aqui com vocês. Me avisem quando...
Já estamos vendo, Júlia.
Já? A apresentação?
Só colocar no modo apresentação, acho que fica maior, pessoal.
Acho que agora parou.
Parou.
Aqui.
Não, eu tô também.
Dá para ver?
Ok.
Muito bem. Então, pessoal, bom dia, eu sou Júlia Spinardi, sou pediatra infectologista e sou responsável pela área médica de vacinas da Pfizer aqui no Brasil. E comigo hoje a gente tem também o Dr. Rodrigo Cini. Cini está por aí?
Sim, estou por aqui. Me escuta?
Sim.
Ok.
O Rodrigo é nosso diretor sênior para vacinas na América Latina. Bom, a ideia hoje é a gente trazer, então, um pouquinho da nossa experiência, alguns casos onde o uso de evidências da vida real, dados da vida real, ou evidências da vida real, foram possíveis de pautar ou ajudar na orientação de algumas conduções dentro do universo de vacinas. É diferente um pouco a perspectiva quando a gente fala de vacina, porque o intuito maior de uma vacina é um impacto especificamente em saúde pública. E para que a gente consiga entender esse impacto, é fundamental que a gente utilize os dados de vida real para entender o alcance de uma determinada vacina numa população. Então, a dinâmica de implementação de um programa de vacinação, ela com certeza se pauta pelo entendimento dos dados de uso desses imunizantes na vida real. Prova disso, a gente vai trazer aqui 2 casos um pouquinho diferentes, de 2 doenças em cenários aparentemente opostos, mas onde ter os dados de vida real, ter as evidências de vida real, podem nos ajudar tanto num cenário a médio e longo prazo, quanto numa decisão mais em tempo real. Então, a gente vai falar primeiro de um caso sobre o uso da vacina meningocócica conjugada na população, para prevenção da doença meningocócica invasiva. Mas vamos fazer um paralelo também com o uso das vacinas contra a Covid, especificamente a vacina da Pfizer contra a Covid-19. E por que a gente fala dessa importância do dado de vida real no âmbito da vacinação? Porque vocês vão ver aqui, pelo caso que a gente vai colocar, que a gente está falando de 2 doenças opostas do ponto de vista de dinâmica. Então, a gente está falando aqui da doença meningocócica invasiva, é uma doença bem conhecida, bem estabelecida do ponto de vista clínico, do que esperar da evolução daquela doença, tem uma alta letalidade atribuída à doença meningocócica invasiva, com sequelas a longo prazo. É uma incidência baixa, muitas vezes alguns especialistas apontam até como uma doença mais rara, mas ela tem uma severidade muito alta, uma progressão clínica muito rápida. Por isso a prevenção através de vacinação é tão importante. Ela se comporta em outbreaks, em surtos, com potencial de casos aparecerem em massa, especialmente em situações de aglomeração. A gente sabe mais ou menos, já bem determinado, quem é o portador assintomático desse patógeno do meningococo, o causador da doença meningocócica invasiva. Então, as estratégias de prevenção podem ser também dirigidas a esse portador assintomático. Apesar disso, a gente entende que existe uma imprevisibilidade da circulação dos tipos de meningococo pelo globo, o que faz com que as medidas de prevenção ampla, ou seja, abrangendo o maior número de sorotipos de meningococo, sorogrupos de meningococo, seja fundamental para as estratégias de prevenção. E tem uma característica, por tudo isso que nós falamos aqui, por ser uma doença de baixa incidência, o desenho de trials para determinar a eficácia dessas vacinas, ele já é bastante comprometido, limitado pela própria característica da doença, né? Então, essa baixa incidência prejudica desenhos de trial de eficácia. Por outro lado, a gente tem um correlato de proteção muito bem estabelecido e que serviu de licenciamento dessas vacinas ao longo do tempo, desde a primeira vacina conjugada, monovalente C, até hoje as vacinas quadrivalentes ACWY para a doença meningocócica invasiva. Então, o uso dados de vida real no cenário da doença meningocócica invasiva faz com que a gente consiga projetar ou entender o impacto dessas vacinas a médio e longo prazo, atuação no estado do portador a longo prazo, implicando num efeito rebanho no restante da corte, mas vamos pensar num cenário onde eu preciso usar o dado de vida real quase que imediato, para guiar as minhas condutas quase que em tempo real. E aí a gente traz aqui aqui o exemplo da doença pelo SARS-CoV-2, a Covid-19. Então, a gente está falando de um novo vírus, com a sua primeira entrada na comunidade, então, com um impacto global, né, do ponto de vista de gerar imunidade para o vírus, né, esse novo entrante, tem uma alta incidência. A severidade da doença vai variar conforme um fator de risco ou uma determinada idade, tem um comportamento pandêmico. A gente sabe que os assintomáticos podem estar em qualquer coorte do grupo etário, não especificamente um grupo é mais portador do que outro. Tem o impacto das variantes, então a forma como esse vírus vai circular nessa comunidade vai fazer com que a gente tenha cada vez mais novas variantes emergindo e pode comprometer tanto impactar a doença quanto impactar a imunidade. Então, eu posso ter um escape imunológico, não estar preparada para lidar com aquela nova variante, como eu posso ter uma doença que se espalha mais ou ter um perfil de maior severidade atribuído eventualmente a alguma variante. Aqui é mais factível a gente desenhar trials de eficácia. Eu estou falando de uma alta incidência, mas a gente precisou adaptar esses trials, os desenhos tradicionais dos trials, para dar celeridade nas análises e, eventualmente, readaptá-los ao longo do curso da pandemia. E a gente ainda tem muitos gaps que a gente precisa entender sobre a doença pelo SARS-CoV-2. Então, qual é o correlato de proteção para essas vacinas? Qual é o mecanismo fisiopatológico da resposta imune? Qual é efetivamente a resposta imune que eu preciso elicitar naquele vacinado para ele estar protegido? Qual é a duração dessa proteção? A gente ainda não entende tudo isso. Então, aqui, o uso do dado de vida real faz com que a gente consiga ter elementos que vão nos ajudar nas decisões quase que em tempo real. A vacinação no mundo real está se comportando dessa forma, precisamos de uma dose de reforço, podemos espaçar as doses, podemos fazer intercambiabilidade, a proteção esperada é de tanto tempo baseado na vacinação em massa, muito antes esse dado vai chegar para a gente do que o nosso acompanhamento de um trial num cenário de pandemia. Eu vou começar explicando um pouquinho mais, então, o caso de meningo, né? Então, as vacinas meningocócicas conjugadas, especificamente aqui nós vamos falar de Nimerix, que é a vacina meningocócica conjugada da Pfizer, protege para 4 sorogrupos de meningococo, A, C, W e Y. Virtualmente, quase toda a doença meningocócica é provocada por esses sorogrupos, né? E como eu falei para vocês, é uma doença de baixa incidência, mas alta severidade, com evolução clínica muito rápida e pode levar, além de letalidade elevada, a sequelas permanentes, né? Então, daí a importância da proteção para a vacina. Quando a gente pensa em estratégia de vacinação para a doença meningocócica, eu já sei quem é o transmissor. Então, a gente sabe que, especificamente, os adolescentes são portadores assintomáticos do meningococo, carregam o meningococo em sua nasofaringe e acabam transmitindo para as outras coortes. Então, hoje a gente entende, e isso veio muito dos dados de utilização dessa vacina nos diferentes programas do mundo, a gente entende que vacinar o adolescente faz parte da estratégia de prevenção de toda a coorte para a doença meningocócica. E, obviamente, eu vacino também os bebês, porque os bebês são os, do ponto de vista de risco, pelo próprio grupo etário, são o maior grupo de risco que a gente tem, as maiores incidências estão na população abaixo de 2 anos, particularmente abaixo de 12 meses de idade. Mas vamos entender como pode o dado de vida real ajudar na progressão de um programa de vacinação. Então, eu entendo mais ou menos a cadeia de transmissão, vamos ver como essa vacina se comporta na massa para eu poder evoluir dentro do meu programa. Vejam aqui o exemplo do Reino Unido, só para vocês entenderem a evolução de um programa de vacinação. O Reino Unido começou a vacinar com vacina meningocócica C monovalente, todas as suas crianças abaixo de 18 meses de idade, num esquema de 3+1, 3 doses antes do primeiro ano de vida e um reforço após completar 12 meses de idade. Com o tempo, ele viu uma redução significativa do número de casos nessa população, teve dados de trial associados à experiência do uso real que possibilitaram a redução do número de doses, passando esse esquema, então, para 2 doses antes de 1 ano de vida, mais uma dose de reforço. Depois, ele segue vendo a queda do número de casos nesse grupo etário, mas entende a importância da vacinação do adolescente como uma forma de inibir a cadeia de transmissão e incorpora, então, uma vacina ao grupo etário dos adolescentes, e isso possibilita com que ele reduza ainda mais o número de doses aplicadas na população menor de 12 meses. Além da população de 13 a 14 anos, ele faz um esquema de vacinar todos os calouros das universidades para reforçar ainda mais esse grupo que é o potencial transmissor, muda essa vacina de uma monovalente para uma vacina quadrivalente, ou seja, amplia o escopo de proteção, eliminando a transmissão de um maior número de sorogrupos para meningo. Isso faz com que eu tenha uma doença ainda estável no grupo etário de infantes, de menores de 18 meses, e possibilite até a prevenção de um outro sorogrupo, meningo B, nessa idade. Então, só para vocês entenderem como e a junção desses dados de vida real associados às experiências, aqui no caso de meningo laboratoriais, possibilitaram que o Reino Unido progredisse no seu plano de imunização e otimizasse o esquema vacinal de toda a população pediátrica e adolescente. Especificamente, esses dados de efetividade de uso de vida real da vacina ACWY vêm sendo cada vez mais mostrados e se tornando robustos com a maior utilização dessas vacinas nos programas nacionais de imunização. Então, nos últimos anos, a gente já vem agregando dados, não só do Reino Unido, da Inglaterra, mas como também da Holanda, da Austrália e do Chile, onde vocês podem ver que os países foram utilizando essa estratégia de vacinação do adolescente e análise do que acontece com as outras coortes para modelar o seu programa de vacinação, otimizando as doses administradas na população pediátrica e aumentando as doses administradas na coorte de transmissores dos adolescentes, que além de, é claro, terem garantido ali a sua proteção individual, passam a ter um papel fundamental na condução ali, no controle de toda a estratégia vacinal. Mais detalhadamente, nós vamos falar um pouquinho sobre os dados da Holanda, onde em 2015 a Holanda enxergou um aumento importante das doenças meningocócicas invasivas pelo sorogrupo W. Tá? Então, em maio de 2018, eles trocaram a vacina meningocócica C por uma vacina ACWY na população menor de 14 meses, porque era a população mais acometida pela doença, então proteção direta, mas alguns meses depois, ainda em 2018, em outubro, eles mudam a estratégia e agregam a vacinação da população adolescente aos 14 anos de idade. No ano seguinte, maio e junho de 2019, eles ampliam essa coorte, vacinam não só os adolescentes com 14 anos, mas os adolescentes de 15 a 18 anos, numa estratégia de catch-up, que a gente chama, com o intuito de não só de reduzir doença nesse grupo, mas de ampliar a capacidade de bloqueio de transmissão da doença para os outros grupos etários. Em 2018, então, a cobertura vacinal nesse grupo etário de 14 anos era em torno de 87%. E o que a gente viu nesse país, o que os dados de vida real mostraram no Programa Nacional de Imunização desse país, nos anos subsequentes, foi uma redução substanciosa da incidência de doença por sorogrupo W, caindo de 0,6 para 0,4 por 100 mil habitantes, ou seja, uma redução de 103 casos para 62 casos. Notem que isso a gente só consegue quando a gente avalia essa vacina na população, para a gente ter esse tipo de desenho num trial. É impraticável, então o uso na vida real é o que vai efetivamente nos mostrar o impacto dessa vacina do ponto de vista de saúde pública, e a gente tem, então, uma redução de 82% na incidência para aqueles pacientes elegíveis para tomar a vacina, mais uma redução de 56% na incidência para aqueles pacientes que não eram elegíveis para tomar a vacina e se beneficiaram do efeito rebanho. Né? E isso em 2019, então vocês veem aqui no nosso gráfico uma redução do número de casos de 18 para 19 e para 20, e uma redução do número de mortes, e em 2020, então, a gente chega a 0 casos no grupo dos 14 anos de idade, mostrando, então, uma efetividade da vacina significativamente importante nesse grupo e um efeito rebanho muito importante que conseguiu estabilizar a transmissão da doença para outros grupos etários. Esses dados foram, então, analisados até março de 2020, vamos ponderar aqui que de março de 2020 para frente, toda a análise epidemiológica tem que ser pautada pelo momento pandêmico, então outros fatores têm o potencial de influenciar a incidência e a prevalência de algumas doenças, mas ainda assim foi um impacto sustentável. Quando vocês olham aqui 2018 para 2019, já uma queda significativa de casos, e para 2020 uma queda maior ainda. É um dado de vida real que a gente só consegue quando utiliza a vacinação em massa, quando a gente consegue enxergar o impacto total daquela vacina numa determinada população. E esse dado serviu, então, para análise junto às agências regulatórias e foi inserido hoje nas bulas, tanto na europeia quanto na bula brasileira, de Nimerix, o que sustenta, né, o que pode auxiliar uma decisão do ponto de vista de saúde pública para determinar uma estratégia vacinal. Então, esses dados da Holanda, mostrando aí uma redução de incidência muito significativa, tanto na população vacinada como nas outras coortes, corrobora e vem auxiliar as agências regulatórias e as autoridades de saúde a ter mais robustez nas suas decisões de estratégia vacinal nos programas de saúde pública. Vamos passar, então, a entender um pouquinho as particularidades de Covid? Rodrigo, você assume daí, eu vou passando os slides aqui.
Tá bom, muito obrigado. Você me escuta bem, Júlia?
Escuto bem.
Perfeito. Bom, muito obrigado pela oportunidade de poder estar aqui falando desse assunto tão em pauta, né, nos diferentes aspectos da nossa sociedade e, obviamente, também em termos de saúde pública. E eu acho uma grande oportunidade poder conversar, poder mostrar esses dados também, justamente após a apresentação da Júlia, que mostra um contexto completamente diferente. Eu acho que um dos primeiros slides que ela apresentou mostrando a diferença entre o contexto da doença meningocócica no mundo e do contexto da Covid-19 é absolutamente diferente. E isso é uma coisa muito importante de se dizer em termos de mundo atual e informações que nós temos, porque a história da vacina é antiga, sabemos disso, mas, por outro lado, também sabemos que muitos dos que nós conseguimos evoluir em termos de vacinação foram nas últimas décadas, em especial, novas tecnologias e possibilidades, obviamente, de novos approaches. Então, isso, obviamente, tem uma diferença grande, tem uma doença já conhecida, tem uma epidemiologia já definida, tem um contexto de saúde já bem definido, completamente diferente quando nós falamos, então, do SARS-CoV-2. Tínhamos uma experiência prévia. Quando olhamos no passado, quando o H1N1 apareceu de uma maneira epidêmica na humanidade, trouxe também muito desta aceleração, desta movimentação em termos de informação. Mas a influenza era um vírus conhecido, não era um vírus completamente novo, né? Ainda que nós conhecemos alguns coronavírus, poucos que atuam na espécie humana, tínhamos pouca experiência. Então, tudo isso nos trouxe um panorama completamente diferente, em que todas as populações são populações que devem ser entendidas e estudadas. O momento, o imediatismo, a necessidade de um combate deve ser bem focado em termos de necessidades, e eu acho que é dentro desse contexto, então, que a gente vai mostrar um pouco do que é a real world data, né, os dados de mundo real, especialmente quando falamos em termos de efetividade. Mas eu acho importante também falar um pouco do contexto das informações científicas necessárias, dos embasamentos que, na verdade, utilizamos nos nossos estudos. E aí, antes de falar um pouquinho dos dados de mundo real, eu acho que temos que falar um pouco dos estudos pivotais, né? Os estudos pivotais, quando, num contexto diferente pandêmico, se faz de uma maneira, ou se fazia de uma maneira diferente, numa aceleração diferente e também numa tensão diferente. O que faz, obviamente, o contexto hoje ser bem diferente e que traz, inclusive, uma série de desafios em termos regulatórios de estudos. Porque os estudos pivotais, como sabemos, eu vou dizer um pouco mais de detalhes sobre esse estudo pivotal, que é o estudo responsável pelo uso de emergência da vacina BRT 162B2, a Comirnaty da Pfizer. Ele foi um estudo que começou também de uma maneira dinâmica. Ele começou de uma maneira com 22 mil participantes, foram crescendo de acordo com as necessidades, aumentando, inclusive, o seu número de sujeitos para mais de 44 mil, baixando, inclusive, as idades de 18 para 16 e até 12 anos de idade. Esse é um contexto completamente diferente, em que, obviamente, todas as atenções estão voltadas a isso. Esse estudo pivotal, como eu falei, foi um estudo multicêntrico, como deve ser os estudos pivotais, tem essa grande valia, e aí a importância em termos regulatórios de conhecermos todos os dados nesse paciente. Esse estudo é nosso, o controle é da Pfizer, da empresa que está, obviamente, conduzindo esse estudo, e isso é fundamental, né? Saber em termos de discussão regulatória, de acompanhamento futuro e entendimento também de todos os aspectos, seja ele do ponto de vista epidemiológico, seja ele do ponto de vista biomolecular. Aqui temos os resultados, então, primordiais dos estudos pivotais. É interessante mostrar que a vacina, com a tecnologia de RNA, se mostrou extremamente efetiva já desde o princípio. O esquema que utilizamos foi o esquema de 2 horas, com intervalo de 21 dias, 3 semanas entre elas, e então mensuramos, avaliamos esses episódios na parte de eficácia 7 dias depois da segunda dose desses casos. É interessante mostrar nesse gráfico que nós temos à esquerda como a vacina respondeu, né, em termos de eficácia. Você pode ver, comparamos as duas linhas azuis, que são dos que receberam placebo, versus a vermelha, que dos pacientes que receberam a vacina. Você pode verificar, inclusive, já a partir da primeira dose, uma boa resposta vacinal que se sustenta e se mantém posteriormente à segunda dose. E é importante mostrar esses resultados, então, de eficácia, né? E podemos aí estar circulados do lado direito, uma eficácia superior ali a 95% e bem robusta em termos de índice de confiança. Então, a partir desse estudo pivotal, que então, obviamente, a vacina começou a ser utilizada, ou foi o pontapé inicial, né, para a utilização em massa da vacina. Os primeiros países a utilizarem foram os Estados Unidos, Israel, os países europeus também. E baseado nesses dados, então, muitos desses países começaram a seguir os seus pacientes e fazendo, então, uma análise mais robusta. Eu acho que essa é a valia dos dados de mundo real, é você trazer um contexto em que aquelas condições de temperatura, pressão controladas nos trials não existem, mas tem uma série de outras variáveis também ali, mas que também nos ajuda e corrobora nesses resultados. O que podemos ver nesse estudo de Israel, que é um estudo observacional por todo o país, que avaliavam a efetividade da vacina, é mostrar um pouquinho dessa história. Se vemos um gráfico à esquerda, podemos verificar todo o início desse estudo epidemiológico, tanto do prévio, ou seja, o que existia até então, né, quando iniciou o lockdown, em que momento, então, se manteve ou se modificou, e em que momento foi introduzido a vacina também, primeira e segunda dose. Então, você tem uma análise aí de um período bem interessante, de janeiro de 24 até abril, 3 de abril de 2021, e se vê, inclusive, dentro da corte que foi vacinada, maiores de 16 anos, uma cobertura vacinal robusta, uma cobertura vacinal maior de 70%, né, dentro desse contexto. E outro fator importante dentro desse contexto, quando falamos, é que, como a Júlia mesmo disse, a doença está em evolução, a doença está em... Pode voltar um pouquinho só, Júlia, desculpa. Quero só concluir aqui. Está em evolução, em contínuo dinamismo. E nisso se vê as diferentes variáveis. Quando estudamos a vacina no estudo pivotal, não tínhamos um contexto de variáveis do SARS-CoV-2 como nós temos hoje. Mas já nesse estudo de mundo real, nós já podemos verificar isso daí. Então, você vê então a presença da variante alfa, que até o final do estudo já representava 94,5% disso, e que também nos ajudava a entender esse contexto frente da resposta da vacina em relação a isso. E o que nós podemos ver no gráfico à direita, então, é exatamente esses valores estimados que vão em linha, inclusive, com os estudos pivotais e que nos ajudam, mais uma vez, a sustentar, a avaliar a importância disso, sem considerar, obviamente, toda a evolução em termos de segurança, que é algo fundamental, que nós estamos aqui discutindo, mas que é um valor que está aí agregado desde o começo. E podemos verificar que uma eficácia superior a 90% se encontra em pelo menos algum desses grupos, né? Inclusive alguns com 96, 97, quando comparamos aos casos graves, que é um fator extremamente importante, porque linha direta com a mortalidade. Seguinte, continuando então nesse dinamismo, vimos o aparecimento de outras variantes, né? De outros contextos também sociais e assim por diante. E aí eu acho que tem esse outro estudo muito interessante publicado através dos dados do Sistema Nacional do Catar, através do seu Programa Nacional de Imunizações, e nesse contexto já tinha, inclusive, essa expansão e essas variantes diferentes, e aí, nesse caso, incluindo dados com a variante beta, que na época era B1351, e vemos, inclusive, dados robustos em relação a dois fatores muito importantes que se discute hoje, né? O primeiro é a infecção, e o segundo é em relação aos casos graves, né, ou fatais, então aqueles casos que necessitam de internação em terapia intensiva e casos de mortalidade. E o que se vê, mesmo que tenha assim, uma variação, e isso é importante também levar em consideração, vou falar um pouquinho no final, que mantém esse número também elevado. Isso é importante, dentro de um contexto epidêmico, principalmente agudo, é muito importante porque causa um impacto imediato social. E aí tem-se que avaliar a longo prazo para a sustentabilidade disso. Vimos que isso responde a alfa, vimos que isso responde a beta, e também um excelente resultado frente a essas diferentes formas de apresentação da doença. Vimos também uma diversidade importante. Isso é outro fator que é importante em dados do mundo real, pode nos trazer também informações complementares. Seguinte. Aqui, na verdade, é apenas um desenho ilustrativo. A ideia é mostrar o quanto que, dentro desse contexto de pandemia, né, dentro desse contexto de necessidade de resposta imediata, os trabalhos, de maneira substancial, acabaram sucedendo em diferentes partes do mundo, né? Para ter uma ideia, nós temos quase 100 estudos aí nesses dois desenhos, e é interessante mostrar, inclusive, a resposta da vacina dentro desse contexto. Nós temos duas tabelas, né? A tabela da esquerda mostrando a primeira dose, a tabela da esquerda após a segunda dose, e você vê que essa robustez que nós vimos desde a primeiro estudo pivotal, e que gradativamente foram apresentados os estudos nacionais, se manteve, trazendo aí uma resposta importante em termos de impacto vacinal. Seguinte. E dentro do contexto que nós vivemos hoje, eu entendo que a doença não está, obviamente, controlada, mas tem essa característica de tentar buscar seu espaço dentro dessas diferentes situações que agora vivemos de controle da doença. E uma dessas variantes que se destaca dentro desse contexto novo é a variante Delta, né? E como a resposta de anticorpos frente a isso é importante em termos de resultados também de eficácia e também em termos de entendimento em relação de uma necessidade ou não de um booster da doença. A resposta imediata, ou seja, os estudos eficazes no primeiro momento se mostraram extremamente promissores. A longo prazo, considerando a doença em expansão, a possibilidade de diferentes variantes, é importante o entendimento também da sustentabilidade desses títulos e também da proteção da população. Frente a isso, seguinte, por favor. Frente a isso, seguimos, então, com os estudos, avaliando os estudos de efetividade vacinal dentro dessas diferentes infecções, e aqui comparamos, então, as infecções delta versus alfa em diferentes estudos, né, que foram feitos nacionalmente de follow-up, estudos de segmento. E aí vale a importância de entender o quanto, né, se pode ganhar com a possibilidade de um booster não a longo prazo e como isso poderia responder. E eu acho que é muito interessante essas três comparações que nós temos aqui. No lado esquerdo temos em verde Espanha, Inglaterra, Escócia e Canadá, e do lado direito de laranja Israel, Catar e Estados Unidos, em que nós temos dois contextos diferentes quanto ao aparecimento da variante alfa e o posterior aparecimento da variante delta. E nesses países em que esse contexto se aproximou, estavam muito próximos, se vê que a efetividade, da vacina se mantém alta, se mantém inclusive bem sustentada com valores acima de 80%. Quando você tem uma variável um pouco maior, e o tempo aí, obviamente, é um influenciador frente a isso, dessas diferentes variantes, você vê, então, uma efetividade diferente, uma diminuição. O estudo de Israel, que foi um estudo, na verdade, inovador e à frente, uma vez que começaram a vacinação primeiro que os demais países ao redor do mundo, se vê uma resposta diferente frente a isso, com valores menores. Inclusive, em outros países como o Catar e, inclusive, os Estados Unidos, que levou a uma discussão da possibilidade ou não da utilização de booster, em que momento, em qual população, em qual maneira, e muito baseado aos dados que eles tinham também em mãos para avaliação. É importante também dizer que, dentro do contexto dos Estados Unidos, a vacinação, para atender números elevados de cobertura vacinal, tardou um pouquinho, então por isso que os dados anteriores se mostravam muito próximos. Mas quando nós olhamos esses dados de efetividade vacinal no mundo real, através de diferentes outcomes no tempo maior de 12 anos, vimos diferentes interpretações em termos de avaliação. Quando avaliamos, então, em termos de hospitalização, vimos que esta variável ela pouco diminui, ela se mantém em termos de efetividade elevada nas diferentes populações em diferentes momentos. Entretanto, quando nós olhamos esta mesma infecção através do tempo, comparado com outros fatores, como a transmissibilidade, vimos que, na verdade, esta variação ela acaba tendo uma diminuição maior e, obviamente, o intervalo entre elas também maior. Então, esses dois gráficos à direita mostram realmente essa diminuição com o passar do tempo, que é, obviamente, muito mais expressiva aqui em termos de hospitalização, e é uma das mostras importantes que nos diz ou pode trazer uma interpretação de que a vacina sim funciona, tanto que os dados em termos de grave e hospitalização se mantêm altos com o passar do tempo, mas que a transmissibilidade vai se diminuir, vai aumentando através e, obviamente, então, a efetividade nesses casos vai diminuindo. Dessa maneira, então, seguinte, é que o FDA, através do seu comitê técnico, VERPAC, de avaliação dessas diferentes situações, considerou todos os dados para avaliar o potencial introdução de booster da vacina utilizando a RNA, a tecnologia de RNA, para essa população americana. E é interessante que essa decisão, através desses dados americanos, deixou bem claro duas coisas. Primeiro, tudo isso é muito dinâmico, realmente isso necessita uma interpretação através do tempo, através de dados e através de informações, aí, através da importância desses dados do mundo real de diferentes perspectivas, e que, frente à realidade americana, se via, sim, a necessidade, isso foi praticamente unânime em termos de votação, da utilização de um booster na população maior de 65 anos de idade, nas populações em risco de infecção pelo SARS-CoV-2, e isso é uma discussão, inclusive, feita posteriormente pela CIP, para que as pessoas não só vulneráveis em relação à doença, mas também que estivessem expostas à doença, seja através do aeroporto, seja através de diferentes funcionalidades, pudessem também receber a vacina, toda essa população acima de 18 anos de idade. E, obviamente, uma vez mais, isso são dados que necessitam de continuidade. A doença, ela segue, obviamente, de diferentes maneiras, e essas interpretações, obviamente, vão ser seguidas para ver se necessita ou não da expansão, mas aí já há um indício claro da necessidade de um booster para um segmento protetivo e uma resposta melhor da efetividade. Seguinte. No Brasil, dentro deste contexto, dentro dessa importância também de dados de mundo real, temos muitos estudos, temos realmente um cenário bem positivo em relação à parte científica. E aqui alguns desses exemplos: estudo no Mato Grosso do Sul, com vacinas de vetores virais, um estudo no Paraná, em Toledo, com a vacina da Pfizer, tecnologia de RNA mensageiro, outro em Serrana, São Paulo, Botucatu, no Espírito Santo, no Rio de Janeiro. Obviamente, dentro de uma realidade pandêmica, de uma realidade de resposta imediata, é fundamental que todos participem e que todos possam contribuir de alguma maneira. Então, não existe apenas uma única tecnologia, uma única resposta de vacinas contra o SARS-CoV-2, e aqui mostra um pouquinho desta realidade brasileira, tentando, obviamente, mitigar, tentando diminuir e melhorar a vida social de todos. Dessa maneira, acho que eu termino a minha apresentação. Muito obrigado. Devolvo a palavra para você, Júlia.
Obrigada, Rodrigo. Encerramos aqui a nossa apresentação de casos, dois cenários bastante diferentes, onde com certeza o uso das evidências geradas no mundo real podem nos ajudar a pensar nas estratégias de vacinação e aumentar o potencial do impacto dessas vacinas num cenário a médio e longo prazo, mas também num cenário quase que em tempo real com relação à evolução da pandemia. Obrigada. Com isso a gente encerra aqui e passo a palavra ao próximo.
Muito obrigada, Júlia e Rodrigo. Passo aqui o próximo, que é o Charles.
Okay, thank you, everybody. This is Charles Lee from AstraZeneca. I will go ahead and share my desktop. Can everybody see? Yes.
No, not yet.
Oh, okay. Let me try again. How's that?
Yes, we can see your email.
Oh. That's the joys of sharing with Zoom. Let me. Right. Okay, how is that? Yeah?
Yes, better.
Yeah.
Okay. Yes, I will. Right. Okay, very good. So, thank you everybody for the invitation to come and speak today about one of the topics that's very central to what we're doing over at AstraZeneca with regards to real-world evidence and real-world data. Sort of building on the studies that we've heard today and in the previous modules, I am going to be presenting a case study of the use of a registry-based study using RWE and RWD for registry purposes. And I'll walk you a little bit through some of our thinking and considerations about that. So in terms of agenda, start just very briefly on the principles around RWE, RWD. I know this has been covered, so I'm going to go very quickly through that. And then also talk a little bit about considerations around pragmatic trials, of which RRCT is one category of that. Do a compare and contrast of how a registry-based study compares with a traditional randomized controlled studies, as well as some of the situations or prerequisites in which the registry-based studies can bring some benefits to patients, to investigators, as well as to industry. And last, I'll close with a real case example of the BEPA-MI study. This is a study that AstraZeneca is currently running, a registry-based study for indication purposes, and I'll talk a little bit about that as well as the regulatory pathway that we took. So this next slide, and I don't need to, I think, explain too much about this already, that you know, because it's been covered, but you know, the distinction between RWE and RWD. The RWD, as the name implies, is the data itself. RWE is the evidence, or the clinical evidence that ultimately is gained and derived from that data, and that's ultimately what is used for regulatory purposes. In terms of potential applications of RWD, now I think there was a very, very similar, almost identical slide earlier today, so I won't talk about this too much. Essentially, we are looking at a spectrum here, and the topic that I am going to discuss today, this registry-based study, is one that sits more or less in the middle of the spectrum between the traditional RCT and using observational cohorts. And I will explain a little bit about why. So in terms of some of the key considerations for pragmatic trials, and back in 2019, there was a workshop by the Margolis Consortium. They've actually held several workshops over the years on RWE and covering different topics of RWE. And here I capture some of the key considerations and the key summary points from the October 2019 workshop. And some of the key points around, you know, in order to support a regulatory decision, the main issue is around the persuasiveness of the findings to provide evidence of efficacy. You know, what we also heard, at least from FDA in several of these workshops, is that observational, so pure observational type studies for indication purposes were unlikely to be accepted in the foreseeable future. And that seems to have been continuing to be reflected as well. I was actually part of a workshop with FDA earlier this year, it was back in February, where it was organized by Pharma, so that's the US version of Interpharma. And AstraZeneca was one of 5 case studies selected by FDA to be presented there. And I think this type of sort of philosophy at FDA around observational studies is still holding. Randomization and blinding are 2 important methods to generate persuasive results, and in the context of RWE are presumed to be very important if we want to provide evidence of efficacy. Ultimately, how pragmatic a trial can be and still be able to provide useful results very much depends on the purpose of the trial and the study question that it seeks to answer, because I think, as we've heard throughout several of these modules and presentations, that there are many different applications, right? For we say RWE in a trial, and what we're using the RWE for, it can be used for a number of different things. At the end of the day, while trials can have pragmatic elements, it is still important to maintain rigorous standards for data collection and assessment, particularly when we are seeking to use the data from these trials, or the results from these trials, to support for regulatory purposes. And in the case I'm going to talk about here, it's around seeking a new indication. Some additional important factors to be considered for pragmatic approaches. And the first one is around sort of the types of interventions in therapeutic areas that might be well suited to clinical care settings. I think we have oftentimes heard of orphan or rare diseases as being one where pragmatic studies or use of RWE can be important. Oncology is again another one. You know, but we also believe there are other areas too, and I'm going to present an example of that later on. An important consideration, and we heard about this earlier as well, is around the quality of the data that can be captured in these settings, and the emphasis on having high quality, robust data for evidence generation is pivotal in terms of using any data for regulatory purposes. And that's also reflected in FDA's recently released guidance as well around data from medical health records and so on. And I think this will be increasingly important, especially as we see digital health becoming more and more prominent, right? These alternative sources of data capture that is a bit different than the traditional that we normally see in randomized studies. And these are going to be important considerations of how we can use those data. The next point, and I touched upon this a little bit as well, is how many patients can be accessed. And again, this is the situation where, you know, in scenarios where the patients are difficult to find or difficult to recruit into a traditional study, you know, the use of pragmatic approaches can be beneficial. Along this same point around data quality, registries are one important data source in many parts of the world, especially in Europe. And as a result, there's a whole EMA registry initiative that's ongoing that aims to facilitate the use of disease registries by introducing and supporting a systemic approach, right, to how the data or the information that's in these registries can contribute to overall benefit risk evaluation of medicines. There was a workshop held, it's almost just about 5 years now, but I think many of the points around EMA patient registries, I think many of the points here are still very valid today. I think in many respects, the registries provide evidence that is very relevant to life cycle assessments of medicinal products in terms of the safety and efficacy in clinical practice. They may actually provide very valuable insights in several ways. They can provide robust data on disease epidemiology, on patient characteristics, as well as the current standard of care and how that changes over time. These information from the registries also represent a source population for traditional randomized control studies, and we see this in practice as well as for data enrichment by contributing an additional data on specific outcomes. The registries themselves also play a key role in enabling pragmatic study trials and post-authorization studies. And again, there are many examples of this where health authorities have been able to have agreed to use of registry type of studies as in the post-authorization setting. It can also provide important information on drug utilization in defined patient groups, or also in broader patient groups as well, because of the limitations around a traditional study. By having access to this data in the registries, it actually provides more of a real world example. So that more or less set the stage for what I'm going to talk about next, which is around the concept of a registry-based study. And what this is it combines elements of the traditional randomized controlled trial using a quality registry data. And what it does is some of the key advantages around a traditional study, because everybody is aware that it includes randomization, causal influence, there's blinding, there's confirmation of efficacy, and last but not least, the randomized controlled trial has long been considered the gold standard for evidence generation. Now, taking those elements and combining those with the benefits of a clinical registry, high quality clinical registry, is that, as I mentioned earlier, much broader patient population that can be targeted compared to a traditional study. Much more effective framework for resource utilization. And I'll talk a little bit in detail about what I mean when I say resource effective, much more efficient type studies for everybody involved. And these registries in many parts of the world are already integrated into normal healthcare. And this is, by incorporating that, it's taking advantage of that to reduce burden. So at the end of the day, the RRCT design, the registry-based study designs, combines all all of these features that I've highlighted of the two other approaches into a much more efficient clinical trial approach while still delivering the high quality data that's needed for evidence generation. So, as I mentioned, the RCT, how does it retain scientific integrity while enhancing effectivity? Well, it really happens in two different areas. One, beginning with the recruitment phase, right? By recruiting patients from a registry, we actually are able to access a much larger patient pool without existing clinical work processes. We have the potential to randomize a larger proportion of eligible patients, right? Much broader patients, right, to increase external validity. Now, also during the trial conduct phase, what you'll see is that this RCT approach minimizes study-specific data collection procedures. It also provides real-time and automatic vital status updates, right, linking to the patient population registries themselves. So some of the key features of an RCT versus a traditional RCT, the top part of this slide shows sort of the layout for a traditional RCT, right? As you can see, there are study visits related to the study itself that the patients have to participate in. But at the same time, these patients also have their routine visits to their physicians. So in many sense, it's increasing both the burden on the patients, the physicians, and the sites. At the same time, when it comes to data collection, there's data that's collected as part of the registries already, just in part of the routine visits. And then there's also additional data captured that are part of the study visits and the patient's participation in the study itself. So you see here a lot of duplication of efforts. In the registry-based RCT scenario, we reduce both of these and we reduce redundancies. One, we combine actually the study visit and the routine visit for the patients and the investigators in the sites. We combine the data collection. In a sense, very little additional data, if any, is conducted in addition to what is already captured as part of the registry. So it reduces the amount of study data that's collected or data specific to the participation of patients in the study is dramatically reduced. And at the end of the day, what this means is that there are fewer study visits for everybody involved. There's much less duplication, almost no duplication of data collection. And this helps to really reduce the burden on patients and much less work, extra work for the physicians as well as the staff that are at the sites. And what this less work means ultimately is that more patients can be recruited. They can be recruited faster at fewer sites and actually at also lower per patient cost, enabling us to do more of these types of studies. Now that being said, our RCTs, or registry-based studies, there are probably certain situations or preconditions, as we call them, that are important or pivotal in order to be able to run and to design and run a successful registry-based studies. And first and foremost, it begins with the registry itself, or registries in this case. Right? There must be a stable and high-quality registry that exists that covers the target patient population that wants to be studied. And also, along the same lines, requires prospectively collected, structured, and regularly validated data entries. And it also needs to have a secure process that that investigators do not lose track of patients. So it really begins with the registry itself. Without a registry, the rest of this type of study approach really cannot happen. The next important consideration is around trial design. So a trial design that should be one that limits investigator burdens. And we've had many discussions for several years on this study before it finally came to fruition around this exact same topic. And we need to be able to design a trial design that is aligned with clinical practice and registry routine outlines. It also means fewer extra visits and to minimize that as much as possible. It also requires an easily captured, straightforward endpoint. So it has to be an endpoint that is normally, for the study, it has to be an endpoint that is normally captured as part of the registry itself, right, to minimize additional data capture. And it needs to be a trial design that requires less extensive safety data collection. And this is related to the point number 3 around the investigational product itself, is that because we want to limit or reduce the amount of safe data in the study, it works best with a drug that has a well-characterized safety and tolerability profile. So oftentimes in situations where we're talking about line extensions, new indications, and so forth. And lastly, in terms of the trial conduct itself, right, there must be measures in place for patient study oversight, and this is directly related to the fact that we're aligning those visits with the routine care, and we need to have certain measures in place to try to make sure that patients aren't lost. So if I go to the next slide, this slide provides an overview of the DAPA-MI RCT design. So this is a study that we are pursuing a new indication for dapagliflozin, for which we are pursuing a new indication for dapagliflozin. Dapagliflozin has been approved in several different diseases, as you are probably aware, beginning with diabetes, but then also now for heart failure, and in the US at least, for chronic kidney disease. So a drug like this has been approved on the market for a long time. There's been a lot of data, safety data that's available on the drug. And because of that, it makes it almost a very good candidate for this type of design. So in this study, we're going to recruit approximately 6,400 patients who have experienced an MI within 7 days. And this study will be done across 2 countries, in Sweden and in the UK, 50 centers within each country, using 2 registries, the Sweetheart Registry and the UK MyNap registry. The primary endpoint here is time to first hospitalization for heart failure or for CV death. So the important point to note here is that it is a hard endpoint. It is an objective endpoint, one that is naturally captured as part of the registries, and really one that does not have ambiguity. That is a very important point. And in the study, these patients will be randomized to either dapagliflozin or placebo. So blinding, their drugs are blinded, there's randomization. 2 important elements of sort of the gold standard that make RCTs the gold standard for evidence generation. And ultimately, it will be an event-driven trial. We expect recruitment to take approximately 18 months, total trial duration of 30 months. Which, when we compare, AstraZeneca has run quite a few cardiovascular outcome studies as traditional RCTs in the past. And what I can tell you is that these timeframes here in terms of recruitment and trial duration are significantly shorter than for an RCT. And the efficiency in terms of resources is also much greater with this type of an approach. Really a close partnership with the clinical centers in the study. A little bit more about that, I think I already talked about most of these, so I won't touch upon the first few points. I do want to touch upon the last few points here, which is around regulatory acceptability of this study to seek a new indication. And we have had several discussions with meetings with FDA, including an end of phase 2 meeting. This is going back to the end of 2019. And subsequent to that, we have received very positive feedback from FDA. We have received fast track designation for this study. We have also received FDA special protocol assessment agreement. And what this is that we submit the final protocol to FDA for this trial. And FDA, if they agree, which they have in this case, it basically means they are agreeing that the study as designed, if the results are and the risk benefit is appropriate, that the design of the study is adequate to support labeling and change in new indication. And this is very important because I think overall, statistically, I think only 5 or 10%, somewhere between 5 to 10% of all submissions for special protocol assessment actually receive the agreement. Oftentimes FDA provides the feedback on the protocol, but to actually secure an agreement is quite a rare event. And this signifies sort of the acceptability of this type of approach to FDA, particularly when, as you notice, the study is not being conducted in the US. and it's actually being conducted in Sweden and in the UK. We've also had scientific advice meetings with multiple EU health authorities in 2020, both local advice with the Swedish MPA, the MHRA in the UK as well, because those are where the patient is recruited, very important discussions. As well as with BfArM, who is the rapporteur for this product, and central advice with EMA, and this happened in 2020. And in general, you know, the feedback from that was the study as designed would be acceptable for the indication, assuming that the benefit risk is adequate. So last but not least, I would like to conclude the RCT that I've talked about today is really a hybrid type of design between a traditional trial and a pragmatic clinical trial. It's one that uses, you know, real world evidence for evidence generation, right, while delivering sort of the characteristics that make traditional RCTs the gold standard for so many years. And because of this, some of the key elements of the RCTs are retained and important in terms of establishing the safety and efficacy profile of the drug. Last but not least, RCTs bring significant advantages to study conduct for both companies, but more importantly, for investigators and patients by reducing their burden to participate in studies. With that, I will conclude and hand it back, I think it's to Alex.
Oi, Charles, muito obrigada pela apresentação. Agora a gente vai abrir para as perguntas que os participantes tiverem. Quem tiver alguma pergunta pode levantar a mãozinha ou pode escrever no chat. A gente agradece a todos os palestrantes. Ela diz, os casos de utilização de real-world evidence e real-world data são importantíssimos. Na minha opinião, ainda falta ter um local onde ter todos os casos registrados. Esse é um comentário. O Leonardo Costa também ele faz uma pergunta, né? Existe uma possibilidade de modelagem matemática, pensando em extrapolação de correlato, de proteção da vacina meningocócica, pensando num possível CP para a vacina da Pfizer?
Não sei se eu entendi completamente a pergunta, mas o correlato de proteção especificamente para a vacina meningocócica, ele já está estabelecido há algumas décadas. Ele se pautou no licenciamento das vacinas meningo-C monovalentes e hoje toda e qualquer vacina meningocócica que se apresente no mercado, à exceção das vacinas de meningo-B, que tem algumas particularidades, mas ainda assim a gente empresta o correlato de proteção, elas devem se provar não inferiores à medicação padrão ouro, que seria a vacina monovalente meningo-C. Então, esse correlato de proteção, ele já está bem estabelecido e a gente consegue estabelecer eficácia pautada nos critérios de não inferioridade para todos os sorogrupos de meningo hoje contemplados por vacina, tá? Não sei se eu respondi.
A Cíntia também faz uma pergunta. Como poderíamos escrever esses casos? Uma publicação em conjunto? E tem uma resposta do Alexander Lead, que pelo menos tem a publicação da ECHO, The Role of Real World Evidence in FDA Approvals, e-book 2021. Isso atende a pergunta, Cíntia? Ok? Bom, pessoal, em virtude do horário, acredito que a gente encerra por aqui. Se aparecerem alguns questionamentos posteriores, pode mandar para a gente, né, da Interfarm, e a gente encaminha para os palestrantes.